Now students, we will discuss allosteric antagonist. So we have already discussed in our previous video that on receptors, there is a second site which is different from that of the agonist binding site, and we are calling this site as allosteric binding site. So what these allosteric antagonists do, they will actually bind themselves to that allosteric site. As a result, there will be downward shift of the Emax. However, there will be no change in the EC50. So they prevent the receptor from activation by the agonist. Here in this case, we have example of picrotoxin, which binds to the GABA receptors. But they are actually binding inside of the GABA receptors. They are binding themselves inside of the GABA chloride channels. So as a result, when picrotoxin is binded or bound to the channel, no chloride can pass or enter into the channel, even when the receptor is fully activated by the GABA. You can see the concept of this GABA receptor from our previous videos. Now, what are the functional and chemical antagonists? Now, functional antagonists, they act on a receptor that is completely separate, thus producing a different effect, different biological response. So these are the agents that may act at completely separate receptor, initiating the effect that are functionally opposite to that of the agonist. Now, let's take an example. We have two drugs, histamine and norepinephrine. Receptor for histamine is different and receptor for epinephrine is different. Histamine binds to H1 histamine receptors, thus causing the bronchial smooth muscle constriction, right? And then we have epinephrine, which is binding itself to the beta-2 adrenoreceptor, and it will act opposite that of the histamine, thus causing the relaxation of the bronchial smooth muscles. So we can say that this type of antagonism is actually a physiologic antagonism. Now, what is chemical antagonism? So these are the chemical agents, these are the chemical antagonists that prevent the action of an agonist by modifying them. So in this case, let's take an example that we have protamine sulfate. This is a chemical and it will act as an antagonist of heparin. Now, this protamine sulfate is a base that is positively charged protein. So it will bind itself to the acidic or the negatively charged heparin. As a result, it will try to affect the heparin. So it will rapidly prevent its therapeutic effect. So this was the example of chemical antagonism. So whenever there is binding of protamine with the heparin, there is binding of positively charged substance with a negatively charged substance, there is production of stable salt. So in this case, we are actually affecting the drug, we are actually affecting the heparin. So in today's lecture, we have discussed that there are agonists which binds to the receptor and produce a biological response. Then we discussed different type of the antagonist. So this was all about the ligands that were agonist and the antagonist.